Welcome to today's From the Journal session, a new event type at the AAAS annual meeting this year, where we invite authors of recently published Science Family Journal work to discuss their research and its implications. My name is Megan Fallon, and I'm the Executive Director of the Science Press Package. Today, we'll be hearing from two authors of the recently published science study, a non-inflammatory mRNA vaccine for treatment of experimental autoimmune encephalomyelitis. The study, which was published on January 8th, reported the design of an mRNA vaccine that delayed the onset of multiple sclerosis-like disease in mice. This is a very timely topic because this work reflects consideration of the mRNA vaccine platform, which we're all hearing so much about related to COVID-19, for a use beyond that disease. This is also work from leaders in the development of one of the two approved COVID-19 vaccines. Here to discuss the results of this research from Science Today are Ugur Sahin, a professor at TRON, or Translational Ecology, at the University Medical Center of the Johannes Gutenberg University, and Ari Weissman, also a professor at the University Medical Center of the Johannes Gutenberg University. They will overview their findings as published in Science, and then I'll be asking them some questions. The speakers will be available to take your questions in a follow-up Zoom room hosted by the AAAS annual meeting. That will be open at 3 p.m. U.S. Eastern Time today, February 8th. You can access that room by going to the meeting's virtual page, virtual.AAAS.org/landing. Now, without further ado, I'd like to introduce our first speaker, Ugur Sahin.
Thank you, Megan. It's a pleasure to have the opportunity to discuss our paper here. So our approach follows the rationale to modulate an autoimmune disease by delivery of a messenger RNA vaccine, which provides the antigen in dendritic cells under non-inflammatory conditions. So these are my disclosures. So let me shortly start to introduce messenger RNA vaccines. So a messenger RNA vaccine is a transcript, a messenger RNA, which is produced by enzymatic in vitro transcription from a DNA template. The DNA template actually encodes, has an open reading frame for an antigen. The messenger RNA itself is very similar to the natural mRNA. It is kept and has structural elements. And usually to deliver a messenger RNA, it is formulated. That means it is packed into lipid, either as a lipid nanoparticle or, as in our study, as a lipoplex formulation. And this formulation protects the mRNA from degradation and allows the uptake of the mRNA by dendritic cells. Once the messenger RNA is taken up by the antigen-presenting cells, it is released in the cytoplasm from the formulation, and then it's translated. And the translated protein is then processed and presented by the antigen-presenting cells on class 1 and class 2 molecules, thereby allowing to stimulate antigen-specific T cells. So in the past 10 years, we developed a number of medical applications using messenger RNA vaccines. Our main field of research was cancer, where we used messenger RNA vaccines in the therapeutic setting, either to stimulate antigen-specific T cells or most recently CAR T cells. We have also established an approach for messenger RNA vaccines for prevention of infectious diseases. But all of these approaches rely on the principle that the mRNA and the formulation together provide strong immune stimulation so that we get an induction of Th1 T cell responses and antibody responses. In the case of autoimmunity, our goal is a completely different one. We want to induce an antigen-specific tolerance, And therefore, in this setting, we started to work with a messenger RNA lipoplex formulation, which provides expression of the antigen under non-inflammatory conditions. So here on this slide, the the technical principles of the work is shown. So we have recently described an mRNA lipoplex formulation, which is injected intravenously, and after intravenous administration, it targets to dendritic cells. This is shown on the left side, where we used a messenger RNA encoding luciferase, and you can see the signal in the spleen region of the injected mice, and when CD11C positive cells are depleted, the signal is lost, showing that the mRNA is taken up and translated in dendritic cells. In this study, we asked the question, what happens if we produce the mRNA in two different ways? On the one side, we use the mRNA, which is prepared by in vitro transcription using standard nucleosides, including UTP. This kind of mRNA is known to stimulate TLR molecules and thereby induce an innate immune response. And we use as a second molecule mRNA, which was produced with 1-methyl pseudouridine, and the incorporation of this nucleoside results in a mRNA that lacks immune activation, particularly if it's purified and contaminants are eliminated. So we injected both mRNAs and evaluated how dendritic cells, which are taking up the mRNA, behave. On the right side, you see that both mRNAs provides a strong luciferase signal, with the nucleoside modified mRNA providing even a stronger signal. But what is very interesting is whereas the normal mRNA induces an immune activation of dendritic cells with upregulation of activation markers on dendritic cells, on T cells, and induces interferon alpha secretion, the non-inflammatory mRNA keeps the dendritic cell profile unchanged. So this visualizes that mRNA expression or nucleoside modified mRNA expression can be used to deliver proteins without inducing inflammation. So we asked the question whether such mRNA can be used for stimulation of T cells. Yeah. So in this experiment, we immunized mice three times, and then evaluated splenic T cells. And we used again here the inflammatory mRNA prepared with uridine, and the nucleoside modified mRNA prepared with pseudouridine. And what we observe is that in both cases, we have an expansion of CD4 T cells. The nucleoside modified mRNA has a higher rate of FOXP3 regulatory T cells, which are expanded. And what is most interesting is in an interferon gamma ELI spot, which measures the stimulation of T cells upon encounter of antigens, here in this case antigen-specific peptides. We see clearly an interferon gamma secretion by the T cells which have been stimulated with the inflammatory mRNA, but a complete lack of interferon gamma secretion by the T cells which have been stimulated with the nucleoside modified mRNA. The same is true for a number of other cytokines. Inflammatory cytokines like interferon gamma, IL-6, IL-12 are not expressed and secreted by the T cells which are stimulated by nucleoside modified mRNA. The only cytokine which showed some levels of secretions are IL-10 and IL-5. So we have induced in this experiment T cells, regulatory T cells with a typical Treg profile, and we lack the expansion of Th1 T cells.
Thank you, Ugo, very much for your presentation, and I will continue from here, and I will discuss some immunological aspects of this study, and also how we think the therapy works in vivo, and I will discuss what could that mean for patients. So as Ugo has mentioned already, the non-inflammatory messenger RNA is delivered mostly to the cells that are called dendritic cells. Dendritic cells are cells that are very important in initiating immune responses. Again, as you heard already, in their trials for COVID-19 and the therapy that is now offered as a vaccine, one needs to activate the immune response to the virus. And for that, you need immunogenicity. This is what dendritic cells do. But in addition to that, dendritic cells, and especially when they are unactivated, and therefore are not expressing all kinds of molecules that they would express when they are activated, they are important in inducing a tolerant state. This is very important in the body, in borders like in the skin, in the gut, and in the lung, where the immune response is constantly encountering different bugs and infections. And there one needs to discriminate between what is a good response and what is a bad response. And therefore, when dendritic cells are in those different borders, they can also induce a state of tolerance. One way that they can do that is by activating and leading to expansion of regulatory T cells. As you have seen already in the presentation by Ugo, we have noticed that when mice are treated with non-inflammatory messenger RNA, we have an expansion of cells that are called regulatory T cells. Those cells are critical for inducing tolerance. People, as well as mice that lack those cells, develop autoimmune diseases. And mice that do not have the critical transcription factor for those cells, that is called FOXP3, would die early after birth from multiple organ inflammation. As you can see here on the left side, when the mice are treated with this non-inflammatory messenger RNA, those cells are expanded. But more importantly, what you can see is those regulatory cells are not only expanded, but they also upregulate the expression of many different markers, proteins that are important for their function, for their good function as regulatory cells. And those includes the markers that are shown here, like TIM3 and TIGIT and LAG3, and also PD1 and CTLA4, that are also used many times as for a treatment against cancer. Because if you treat the cancer patients with antibodies against these markers, you will get the regulatory cells inactive and therefore will allow effector function. This is an example, not from our paper, of how the disease, the experimental autoimmune encephalomyelitis, or EAE, is used as a model for multiple sclerosis. The animals are immunized, in our case, with the peptide done from MOG, together with a strong adjuvant. And a few days after they are immunized, they will develop paralysis that will be temporary, and then slowly they will recover, as you can see here, in comparison to the flat line of the control animals unimmunized. What you can see here is from our paper. So when one treats the animals after the induction of the disease, but before the signs of the disease with the non-inflammatory messenger RNA, the mice, what you can see here in the blue color, are completely resistant to the disease, and you can see the control animals get sick. This can be seen, as I said, by the phenotype of the animals, but you can also look in the central nervous system itself, in this case, in the spinal cords. And if you look at the spinal cord, you can see in dark blue or violet color, you can see the myelin. And you can see the myelin is intact in the animals that were treated with the non-inflammatory RNA, because those mice did not get sick, while in the controls, the myelin is lost. But not only that you can prevent the disease, but you can also treat ongoing disease. If we wait now until the mice start to develop the disease and only then give the non-inflammatory RNA, we can see that the animals, again here shown in blue, recover much faster than the control animals. So not only prevent the disease, but also treat ongoing disease. But the biggest promise in my eyes in this treatment is that one can potentially also treat against responses that are not yet known. So what was done here was that animals, first of all, another mouse strain to show that it works not only in one strain. In the case I've shown you the data until now, these are C57 black 6 mice, but also in this strain that is called SJL, with another antigen, with a PLP antigen, you can induce the disease, and you can also prevent the disease if you treat them with non-inflammatory RNA that codes for this PLP. This is all the same like was done for the MOG, with the MOG, the RNA, the non-inflammatory RNA coding for the MOG. But what is important here is that not only the PLP RNA was used to treat, but also the MOG one, although this was not the one used for the induction of the disease. And this is a bystander effect. What does it mean? It means that the treatment with the messenger RNA that codes for the MOG protein was enough to also block the response against the PLP. And this comes most likely because the expansion of the regulatory cells that are specific to PLP were also enough to tolerize against the T cells that were specific to MOG. And this is called bystander effect, which would potentially means that we will be able to tolerize against antigens that we are not even sure what they are, as long as we can activate the regulatory cells in the right place, in our case of multiple sclerosis, in the brain and the spinal cord, and in the right time. So to summarize it, multiple sclerosis patients have effector T cells with specificity to many brain antigens, some of them not yet identified. Treatment of those patients with therapies aimed at the effector cells, not the regulatory cells, but the effector cells so far failed. This could be because the treatment were too radical and therefore led to other problems in those patients because they had not sufficient immunity, or they were not sufficient. The treatment that we are offering here would lead to the expansion of Treg cells that have the promise of generating bystander effect. And as long as the regulatory T cells are activated in the right place and in the right time, they could inactivate different effector cells. As you can see on the right side, an example of such a patient that could be potentially treated with this vaccine messenger RNA. And thank you very much for your attention.
Thank you both, Ari and Ugur. That was very insightful. I want to remind everyone in the audience that you are going to get a chance to ask questions of these speakers at the follow-up session at 3 p.m. Eastern Time. And now I will ask a few questions of our speakers. And Ugur, I'd like to start with you and ask a big picture question. Backing up from this work in science on MS to your role in delivering an approved mRNA vaccine for COVID, I just watched a 60 Minutes interview that you had done where you talked about this work. And what struck me was your saying, early on in the pandemic, what concerned me was we might be too late. But now we're here, it's January 2021, we have 2 approved mRNA-based COVID vaccines. In your mind, what did 2020 show us about what science can do on a dime in a crisis?
Yeah, it is indeed an extremely interesting situation. So we have 2 approved vaccines, and it is the fastest development for a vaccine against a novel pathogen in the history of medicine. And the experience shows us what is possible if we work together, yeah, and if we make our decisions on a pure scientific approach. We had the privilege to be able to rely on more than 20 years of research work. But usually a development of an infectious disease vaccine takes several years. We reduced that to less than 1 year, and this was only possible because we reduced the time and eliminated everything which is unnecessary. Waiting times, decision making was done fast. Investments were made before getting the results. So this is an extraordinary situation, but it gives us also it provides also the question whether this type of developments could be also possible for other type of diseases where a high medical need is existing, and where we know that development of new therapeutics requires many years, sometimes more than 10 years. So the question here is, of course, can we shorten the development times for innovations?
That's a great segue. Your mention of applying this approach to other diseases to the next big picture question I want to ask before we come back to the work published in Science. Ari, I wanted to ask you, we have 2 authorized mRNA vaccines for COVID-19. The mRNA vaccine approach uses a lipid nanoparticle to deliver the mRNA. How long does the mRNA stay in the body after it's delivered like that? And is the time in which it's there any kind of concern for use of this vaccine approach in other diseases where potentially you'd need it to be there longer or shorter to mount an immune response that's appropriate?
Thank you for this great question. I think the timing is very difficult to answer, but I think it's probably the right timing. First of all, dendritic cells have a very short half-life anyway, so it wouldn't help to have the messenger RNA hanging around for much longer. Secondly, once those regulatory T cells are formed and expanded and activated, they will hang around for a very long time. So what the messenger RNA does in a relatively short time will be sufficient to have the treatment for a long time.
Very helpful. Thank you, Ari. Ugur, so you published this paper in Science on January 8th, about a month ago, but this is showing this mRNA vaccine approach in another disease, one not caused by a virus like COVID-19 is, but one caused by the immune system attacking itself, multiple sclerosis. And you reported the results in the mouse study, as you both have described. First, I want to ask you, why MS? Why is that the disease that you started with here with this approach?
It's a combination of factors. First of all, it's a disease with a high medical need. Even though a number of different treatments are applied, this kind of treatments come with often with limited disease control and with the side effects of systemic immunosuppression. The second aspect is there is a fantastic scientific field around MS research. The models are well established, the immunology is well known, There are autoantigens which are accepted by the community as relevant ones, and we have also a path for potential clinical translation, because also in the human disease there are a number of well-known, well-characterized autoantigens which could be applicable, yeah, for as targets for such type of messenger RNA vaccines.
So Ari, you tested this vaccine approach in mice. What are your next questions about using this vaccine for this disease, and how will you go about exploring them?
So first of all, about the initiation of the project, I think it's also important to mention that we have done the ground work on this project originally using genetically modified animals, because my lab does genetically modified animals. But we couldn't move further on because you need to have a therapy for people. And of course, genetically modified animals are nice for showing principles, but they are not a therapy. So this opportunity to collaborate with Ugo and his team allowed us eventually to reach to a situation where a therapy could be at hand eventually. And I'm sorry, your question, yes, what are the next steps? So I think that for me as a person that is mostly interested in mechanisms, so at least that's what I was interested until now in my scientific life, I would like to understand a little bit better about how does that work. And also, can we also target this vaccine more specifically to the brain? So in the brain, you have other cells that are antigen presenting cells. You have also dendritic cells, but you have other cells that are more specific to the central nervous system. Can we actually target this vaccine to them and leave the peripheral organs out of that? So those kind of questions, and understanding a little bit better the mechanism of how to get those regulatory cells, maybe make more of them and more specific, for example, by allowing the dendritic cells also to produce the cytokines we had in Ugo's talk, IL-10, TGF-beta, that will possibly give us more regulatory cells, and that could be also more specific.
Look forward to seeing that research, hopefully in Science. Okay, so Ugo, a question for you. Thinking about extending this approach beyond COVID-19 and multiple sclerosis, how could mRNA vaccines be used to make universal flu vaccines, which are so sought after, better, where current approaches haven't been able to do this?
Yeah, so in principle, the great power of messenger RNA vaccines is that we can precisely engineer the mRNA, allowing us to express the protein, or to get the protein expressed by human cells in the conformation that is needed. With regard to universal flu vaccines, this could be engineered stalk proteins, and also applications which could be innovative prime boost protocols. But what is also important is that beyond the idea of a universal flu vaccine, the flu vaccination can be improved itself by providing a vaccine which is better tailored to the actual flu strains. The key advantage of a messenger RNA is that the mRNA vaccine could be available within 6 weeks. Yeah? So that means we could use information which is much more accurate about the actual flu strains to make a vaccine, and that could already make a difference and a better vaccine.
Just to follow up, how does that 6-week time frame compare to what is needed now to identify and put into place the strains for the flu vaccine?
This is more in the time range of 6 months. Yeah.
Okay, a terrific advance. Ari, What about using this approach to fight cancer or HIV, places where researchers have spent a long time working on vaccines with only modest success?
Well, first of all, as was already mentioned, here we are trying to achieve the opposite from what one tries to achieve in cancer or infection. We are trying to get the regulatory cells more potent, more and more potent. And in the case of cancer, what we want is to have less of them and better effector responses. Now, actually, this question would be best addressed to Ugo, but I can try to answer for him. So this technology was developed by Tron, the institute that is led by Ugo, in order to develop vaccines to cancer. And I think that they are getting there. They are very advanced in that. And from my point of view, I don't know what Ugo thinks, but the fact that now you actually could show that you can deliver this vaccine that is very sensitive to temperature, but that you can deliver it. I grew up in Israel, and Israel has something like 2 million vaccinated people already, so that you can have a setup to deliver this vaccine so efficiently will allow now to see the promise of that also for other diseases like for cancer.
Thank you, Ari.
And you asked me about HIV as well. Would be the same direction, only that, as we know, HIV changes much more than does COVID, and actually what Ugo mentioned before, that it allows us to change the vaccine very quickly, could have actually a great promise for HIV.
And Ugur, did you want to add any of that, your perspective from working and leading at Tron?
Yeah, I fully agree with Ari said. I would define as a vaccine as a way of talking to antigen-specific immune cells and telling them what they should do. And every time when we have a disease which can be modified by antigen-specific immune responses, we should consider a vaccine as a potential treatment or a way of preventing it.
Okay. Now I have one more question before I'll move to our lightning round question for both of our speakers. This is reflecting a concern. We've seen a lot in the news about variants of the coronavirus. One is circulating in the United Kingdom, causing a lot of disease and harm. You were an author on a preprint that was recently published, Ugur. It talked about patients who'd been given the Pfizer-BioNTech vaccine. The blood appeared to neutralize the variant of the coronavirus just as well as the normal form of the pathogen, if you will. That seems like good news. How will you need to continue to study the ability of this vaccine to work against this and other variants?
Yeah, so we studied the UK variant and have observed almost equal neutralization, and we will continue to study other variants, the South African, the Brazil variant, and other emerging variants. So this is important that we get an understanding of the evolution of the virus and how the new variants might escape or not escape vaccine responses. I'm confident that the vaccines that have been approved, because they are inducing not only antibody but also T-cell responses, will not be much affected by this type of variants. But it is important to continue to monitor not only the molecular epidemiology, but also whether variants induce more severe disease to inform us whether new vaccine variants would be needed or not.
That's hopeful news, and we'll continue to monitor, as you say. Okay, So before I close, I have a lightning round question, one for both of our speakers. And we'll start with Ari. And the question is, how is advancing this mRNA vaccine approach that we've talked about so much going to offer benefits to patients that other therapies would not?
Well, I think that it's early days, first of all, that's very important to say. But what this will be probably cost effective, because as we have seen, this vaccine is not very expensive, and the treatment for multiple sclerosis patients so far are very expensive. And secondly, we have a lot of treatments for MS patients, but in most of them, we actually do not understand why do they help to the patients. And here we understand exactly.
Thank you. Ugur?
Yeah, it is extremely important to develop such type of novel treatments in a scientifically accurate and diligent way. The development of medicines requires a good understanding of the mechanisms, and also requires biomarker analysis and long clinical follow-up. Therefore, this type of novel treatments will not be available tomorrow. It will take years until such innovations could be made available to patients on a regular basis.
Thank you. We've been speaking today with Ari Weissman and Ugur Sahin. They've given us terrific insights into the mRNA vaccine approach that's being used for COVID-19, and also how it was applied in a mouse study published in Science looking at prevention of a multiple sclerosis-like disease. I want to thank both of our speakers for their insights today. They will also be available in a follow-up Zoom room, as noted, at 3 p.m. Eastern Time, so in just a few moments. And their paper is available on the Science Magazine website. We thank our speakers and our attendees for coming to this session of the AAAS annual meeting.